Thank you, Minji and Kim Fai for the invitation, and thanks, Jun, for the kind introduction. So I am now at the company. The name of the company is GV20 Therapeutics. So some of you might wonder whether this has anything to do with Google Venture. Unfortunately, it doesn't. When we started the company in 2016, the core technology was CRISPR screens. My lab started using CRISPR screens for oncology and immuno-oncology studies in 2014. And we also developed many algorithms for developing more efficient CRISPR screens and for analyzing the high-throughput sequencing data to get hits from CRISPR screens. And so when we started this company, the Broad Institute CRISPR screen technology platform is called Achilles Heel Project. So we were trying to see whether there's another name we can say like cancer vulnerability. So in the Chinese acupuncture point, there are some acupuncture point on the back of the spine, like on the spine. From the tailbone is GV1 and 2 and 3, all the way up, and on the top of the head is GV20. 百会穴. So we are trying to use genomics and computation to find cancer vulnerabilities. So let me start the talk with a question. Do you know the top-selling cancer drug in 2022? Yeah, it's an anti-PD-1 drug from Merck, right? Keytruda, right? It's the number one. Of course, BMS also sell a very similar drug, Vivo, and Roche sells a related drug, anti-PD-L1. And so together, these three drugs sold for over $35 billion in revenue. So what are the conventional approach to discover a drug like an anti-PD-1 or anti-PD-L1? The process usually works like this. It's from the target to the mechanism to the drug. So first you have to know there's a gene called the PD-1. You need to understand its function or the mechanism. So PD-1 is a gene that's highly expressed on the T cells, and it interacts with PD-L1 on the antigen presenting cells. And this pair of interaction makes the T cell in a dysfunctional state. Therefore, you can develop an antibody either against the PD-1 or PD-L1 to block this interaction. And therefore, then when the T cells are activated, when the T cell receptor recognizes the cancer cell as non-self, then the PD-1 antibody can maintain T cell in a constant activated stage or maintain the energy, the activity of the T cells. So overall, this is a pretty long and expensive process. You have to understand the gene, know its function before you can develop a drug. But do you know that cancer patients in their tumors already make anti-PD-1, anti-PD-L1 antibodies? This is actually the hypothesis when we are taking to developing the company. And so in recent years, there are increasing realization in the tumors, there are small lymph nodes. And so in there, the tumor, the dendritic cells are presenting the tumor antigen to our immune system. And so this tertiary lymphoid structure, these tiny lymph nodes are proliferating T cells and B cells against the tumor antigen. Especially we're interested in B cells. So these B cells in the patient tumors, in these little lymph nodes, they can recognize the relevant tumor antigen. It can get activated and then gradually evolve to optimize antibodies against the tumor antigen and then mount attack against the tumor. Unfortunately, even though some of these patients are making perfectly good anti-tumor antibodies, they are insufficient to really defeat the tumors. There are two reasons for this. The first is that they don't make enough. So you can imagine when you have a war, just having a small group of good soldiers are insufficient. You really need a whole army of good soldiers. So that the amount is one thing. The second is that when the antibody is still on the B cells or on the plasma cells, the B cells themselves can still go through a process to shift the Fc end. So if you look at the antibody, the Fab region recognize the tumor antigen, but the Fc region, they call out a different immune help. So when the antibody is in a B cell, it's in a naive B cell, it makes IgM antibody. When it's activated, it makes an IgG antibody. But then the cytokine environment in the tumor can induce the B cell to shift from an IgG antibody to an IgA antibody. And we believe once the antibody is an IgA antibody, it's no longer tumor killing. It's actually even tumor protective. So our hypothesis was that normally IgA antibody actually appears in the mucosal tissues, like your airway and your GI tract. Because the air you breathe from outside, the food you eat from outside clearly contain non-self things. And there's no point to get your alarm system, to get your immune system alarmed against these food and attack them. And so once we hypothesize this IgA antibody that's lining your gut tissue is actually suppressing the nearby immune system. And therefore, when the cancer cell in a tumor, cancer cell never do anything de novo. They always learn something from stem cells, from other cells. And so cancer cell realize that if it's actually producing the cytokine, the B cells can shift the antibody from IgG to an IgA. And then this piece of tumor will look like a piece of food to the immune system. And then your immune system wouldn't attack it, right? You become pro-tumor. So the way we came up with the hypothesis was to look at the TCGA data. So in the amount of B cells in the tumor, usually if you have more B cells is good, but the percentage of IgA antibody is actually bad. When you have more IgA, the patient actually do worse. We then also checked the literature. There was some earlier work from Michael Karim's lab to show this, and later on there was a spatial transcriptomics work from Detmer Wolf's group. They also show that basically the B cells are matured in this tertiary lymphoid structure, and then through the fibroblast tract, it will migrate to the tumor niche. And if the B cells going to that location is producing IgG antibody like this one, then the adjacent cancer cells are more apoptotic. Whereas if it's producing IgA antibodies, then the nearby cancer cells are no longer being killed by the B cells and the antibodies. And so then we thought, oh, okay, so the patient are trying, they're making antibody, but they're not making enough. And then very quickly, the tumor also is forcing the B cell to shift the FC to the wrong FC, then it stops killing the tumor. Therefore, what if we just directly give patient antibodies? Because if you give antibody as a protein, it cannot actually shift FC. It only happens on the B cells. So if we give the patient enough of the antibody with the right dose, with the right format, then this could be very powerful therapeutics. And that's kind of the approach GV20 is taking to develop our therapeutics. Where do we get these antibodies? We are getting them from tumor profiles, RNA-seq profiles. So nowadays, it only takes $200–300 to do the RNA-seq of a tumor. And when you sequence the RNA, you don't just sequence the cancerous cells, you also sequence the infiltrating B cells in there. And the B cells are also making the RNA that eventually produce the or translate into the antibody. The only thing is that in order, like B cells have only 2 copies of the DNA, but it can produce, billions of different type of antibodies because it goes through a VDJ recombination process. But once it creates this hypervariable sequence to recognize potential antigens, then it's no longer mappable to the reference genome. And so when people do RNA-seq, the very first step of data analysis is to look at the RNA-seq reads and try to map them to the reference genome. And all the antibody sequences get thrown out as unmappable garbage. As a computational biologist, we take pride in collecting or in picking out gold from garbage. And so we were thinking, this VDJ recombination, when it's translated into protein, the V sequence and J sequence, they have some amino acid sequence patterns. And so we try to look at all those unmappable reads and try to translate them into different frames and ask which one of them contains some patterns that they are coming from a B cell. from the V or J sequence? And if so, especially for paradigm sequence, you might have one end that mapped to either V or J, the other end is like unmappable. Then we try to kind of assemble them together by overlapping the reads. And so we can then assemble out the sequence into antibodies. On average, from each tumor RNA-seq, say in TCGA, we can get a few thousand antibodies from the tumors. So we did a comparison to say, well, what if we just do tumor BCR sequencing? This is actually a pretty expensive assay. To do BCR sequencing is probably like $1,000. But you can also do RNA sequencing, then run this trust algorithm that we developed. And so we saw that we are not able to assemble all the BCR sequence. Instead, we are getting the really abundant ones that are clonal expanded, therefore are more likely to be recognizing tumor antigens. And so then in 2019, we published this paper in Nature Genetics. This was actually dealing with bulk tumor profiles. Since then, we have published another study. Even if you do single cell RNA-seq, but without specifically amplifying the VDJ region, if you're using the 5' 10x genomics kit, just a regular 10x study, we can still use this trust algorithm to assemble the TCR and the BCR sequence from the single cell data. Anyhow, the first author of the 2019 paper decided to join the company. So we started collecting tumor profiles and then just run this trust algorithm. And so we have like tens of thousands of tumors, and then we have over 100 million antibodies that we computationally extracted from the patient tumors. And so we were kind of thinking, what are we going to do with these antibodies? And, so one day we thought, oh, maybe we can use AI to predict the targets for these antibodies. Also, we later also realized using this approach, we can tell which target are potentially more disease relevant, and in this case, cancer relevant. And so later on, we call this approach simultaneous target evaluation and antibody discovery. So let me intuitively explain to you roughly how we use AI to make the predictions. Let's think about image analysis. AI has really played a very big role in recent years. So in order for AI from, say, Google to differentiate the image from a dog or a cat or tiger or a lion, what you need to do is to feed this AI labeled individual pictures of cats and dogs and so on. And you have to have sufficient number of pictures, right, from different angles, from different individual animals. And so over time, the AI can automatically learn the features that can tell dog apart from a cat or a lion. And so this is automatically learned. And so for us, this is actually a pretty easy question. We are dealing with a much, much harder question. So imagine you have a zoo, and before you go to a zoo, you go to the website and you can see, say, London Zoo have the following animals, Tokyo Zoo have different animals, San Diego Zoo have different animals, and so on. Supposedly on the website, they don't tell you what the animals look like. You just know, okay, the zoo has these animals. What you can look at are pictures taken by tourists who visit that zoo. And they don't specifically label what animal each picture belongs to. But at least you know that these pictures coming from the London Zoo are all pictures for the animals in that zoo. If we only look at a single zoo, we have no way of telling what animals look like, which type. However, if you have this unlabeled picture, but a sufficient number of zoos that are more or less, very different, then what you can say is, well, let's look at all the zoos that have lions, and then look at all their pictures. Do they have something in common, which are absent in the zoos that don't have lions? How about tigers? Look at all the zoos that have tigers. What do they have in common that's absent in the zoos that don't have tigers? How about zebra, panda, and like every animal? And so at the end, if you are learning this correctly, all the animals need to match on all the zoos you have, right? Then you know that this AI is working. But for us to learn, to know that we are learning this correctly, at least in machine learning, is by cross-validation. Imagine we leave some zoos out, say Zimbabwe Zoo. We have never seen this. We finish all the training with all the rest of the zoos, and then we finish the training, we ask, give me the pictures from tourists coming from Zimbabwe Zoo and say, if based on the pictures, we can say, well, they seem to have zebra, giraffe, and hippo, but then they don't have panda and penguin. And if this actually agree with the animal in the zoo, the list, then we know we are learning correctly. And so basically the AI can automatically learn some features to tell animals apart from the other without ahead of time individually labeled pictures, right? So this is kind of de novo learning. You don't really need labeled pictures, which is pretty nifty. And so in our case, every tumor is a zoo. And in each tumor, you have many antigens. It's like every zoo have many, many different animals. And in each tumor, we also computationally extracted many, many antibodies. And they are like the pictures in that zoo. And so we know that within each tumor, the antibodies are supposed to only recognize the antigens in that tumor. And so if I have only one tumor, I can't tell how to match them. But if I have so many different tumors and they're all different, I can do the same thing, de novo infer the antigen antibody pairs without knowing a database of antigen and antibody in a database. So the basic idea is that when you see a target being present in the tumor, do we see some antibody sequence features that repeatedly are associated with this target? And they are absent in the tumor that don't have this target, right? So the nice thing there is we are not asking the whole antibody sequence to be identical in all the patients. Because when the antibody sequence could be pretty long, the actual binding sequence is called the paratope. They could be just 6 or 8 amino acid long. And there could even be short patches with a gap in the middle. The AI can automatically figure this out. What are the important features that are repeatedly appearing with that target in the patient tumors? And that's kind of the beauty of this. It's like you are not asking the whole tiger to look the same. Instead, you're looking at some, patch on the eye, on the skin, and on the nose, then oh, this is a tiger, right? So we are doing kind of the same thing here. And so another interesting thing is when we see an antigen in the tumor, and if many, many patients repeatedly make antibody against that target, it would tell us that this target is potentially a good cancer drug target. And the antibodies that we already saw in the patient tumors are potential good drugs that we can develop into antibody therapeutics. So an analogy of this is like this. You can imagine in a zoo, when the zoo have a panda, tourists love to take photos of this because it's really cool. They share it on their social media. So panda is very photogenic. Whereas if the zoo has a goat, it's so boring. Who wants to take photos and generate and share it on their social media? Then it's not as good a target. And so based on the pictures, we can tell which target is, or the antibodies, we can tell which target is more interesting. So actually, based on the antibody signals, we could already tell PD-1 is a very good target. Interestingly, many patients make a little bit of PD-1 antibody already in their tumor. It's a better target than PD-L1, and it's actually much better than targets like CD47 and Siglec-15. These two drugs, were developed in the clinic and showed a lot of promise initially, but we almost never see patients making antibodies against them. And unfortunately, they're not doing well in the clinic right now. And so I can show you the PD-1 result. So without looking at any existing PD-1 antibodies, we just totally use the tumor profiles to help us build a de novo PD-1 model. And with that model, basically, after we finish the AI, if you feed the AI model a sequence, the AI model can tell us whether this is going to be a PD-1 antibody or not. And so there was a paper published where they synthesized 10 PD-1 antibodies that are already in the clinic. And they individually measured the affinity of these antibodies against the PD-1 target. And so we ran those 10 sequences into this de novo derived AI model against PD-1. And not only we can predict that they are PD-1 antibodies, but also our predicted affinity, surprising to us, even correlated with the measured PD-1 affinity. So the early PD-1 antibodies developed by Merck and Bristol Myers Squibb, they actually have pretty low affinity. The later antibodies developed by Novartis and also BeiGene, they have higher affinity, which is not surprising. You know, a lot of times, the later companies they just do this. They say, okay, there is a PD-1 antibody from Merck against the PD-1. Let's develop another antibody that can kind of squeeze out the Keytruda antibody. Then it certainly would have a higher affinity. And so we ask, can we use AI to generate another antibody? This is, by the way, it's not really a generative model. We are just pulling out antibodies that are already present in the patient tumors. And so then we tested them. Indeed, they bind. But interestingly, it binds with very high affinity, but it's a different epitope with Keytruda. They don't really compete with Keytruda, but we know it can fully block a PD-1, PD-L1 interaction. And so when we tested this in mouse tumors, so mouse and human have different PD-1 sequence, and the functional domain is different. So we have to get a mouse that expresses a human PD-1 to test our antibody and the Keytruda antibody. And you can see that our antibody actually even works better than the Keytruda antibody in terms of tumor control. Of course, we're not developing this antibody because Keytruda antibody will be out of patent in 2 to 3 years. And so this just serves as a proof of concept. And so with this, we kind of gain more confidence. We ask, okay, can we get other antibody? You know, we tried, can we get EGFR antibodies? Yes, patient make EGFR. Can we get HER2 antibodies? Yes, actually there is a boatload of HER2 antibodies in patient tumors. We also tried other either like pretty hot targets in immuno-oncology or oncology like ADC targets. And in each of them, we were able to use AI to pull out antibodies from patient tumors that we then validated indeed bind to the target of interest. And some of them have very, very interesting properties. So for example, you know, the B7H3 target is actually very conserved between human and mouse. And if you were to use this target as an antigen to immunize the mouse, at the end, most of the antibodies generated are only against the region that's different between human and mouse. Whereas, you know, when we get this antibody, it's actually cross-react to both human and mouse, which is great. And there are also other antibodies like this one against Nectin-4. The initial AI predicted antibody we found can preferentially bind to Nectin-4 in an acidic environment. We hypothesized that in the tumors, it's actually already very acidic because of hypoxia. So the tumor is actually more acidic. And so the antibodies that are generated under the natural environment just bind to the acidic form in a higher affinity than the neutral form. So we feel like because we're deriving these antibodies directly from patient tumors, they kind of have all these tumor properties as well. And so here is the way we kind of use the platform for our drug discovery effort. Again, we call this platform simultaneous target evaluation and antibody discovery, because most AI drug discovery companies, they need to use either published literature or pharma partnership to nominate a target. And the function of the target needs to be fully understood before they can use AI to develop drugs. But in our case, we kind of don't do it this way. We're using 40,000 cancer patients as our experimental system. And we know that they're all trying, right, to cure the tumor. They just failed, but at least we can see what kind of battle do they fight. And if there are some local battles that are repeatedly fought by many, many patients, maybe that's a good battle. And so based on the tumor profile and this AI antibody matching to the target, we can already kind of rank, you know, which is panda, which is goat in terms of cancer target. And each of them, we use AI to predict which are the corresponding antibodies against the target of interest. So that's kind of our initial ranking of which gene are interesting. And the second step is really prioritization. Remember, we started the company as a CRISPR screen company. So in the first 3 years, we did many immune-related CRISPR screens. We just knock out each gene in turn in a cell population, and then see which gene knockout give us the phenotype, say tumor growth or cell surface protein expression and so on. We also collected a lot of public CRISPR screens, and we really asked, you know, which gene knockout give us an interesting phenotype. The rest is just more public data mining. We look at normal tissues, cancer cell lines, mouse tumors, and human tumors, and to look at DNA, RNA, epigenetics, proteomics data, some single cell data, and also very limited immunotherapy trial samples to help us generate hypotheses. Is this gene expressed on cancer cells or immune cells? Is it associated with some immune signal? Is it influencing patient survival or immunotherapy response, right? So with these CRISPR screens and public data to help us prioritize, we then go to the experimental validation, ask if the AI predicted antibody indeed bind to the target of interest, and then in vitro or in vivo characterize the antibody. And that's how we came to the lead program. And so in terms of other AI, especially AI antibody discovery companies, there are many companies recently. Some of them, they don't come up with novel target. Also, many of them will require a known antibody against a target already. So if you already know this antibody binds to this target, and then you can experimentally generate a lot of slightly different sequences, and you can test whether that makes the antibody bind stronger or weaker. And with enough data, you can build AI models to get an even better antibody. For us, that's kind of a pretty simple, easy question, right? There are also two companies, they are getting antibodies from patient tumors, but they are sequencing the tumors themselves. And so from like a dozen patients here and there, and they're asking the full-length antibody to repeatedly appear in 2 patients or 3 patients. It's much, much harder. And then once they have an antibody, they test it on a protein array with all like 10,000 different antigens to see where this antibody might bind, which is pretty cumbersome. Especially say if you get one HER2 antibody and it has some issues, you say, oh, give me another HER2 antibody. You will take the many experimental testing to get you a second, because every time they try a random antibody, they hope it's a HER2, right? It's not easy. But in our case, we start from many patient samples. And we use AI to already say, okay, this target is potentially interesting. And every time when we make a prediction, we predict many antibody against that target at the same time. Another thing we found is that if this paratope is really repeatedly appearing in so many patients, it seemed to be enriched in functional binding. Say on the PD-1, there are many different locations you can bind, but the functional antibody can really block PD-1, PD-L1 interaction. If this thing is functional, then that antibody's production will stimulate some local cytokines, allowing this little B cell to expand even more, or also repeatedly in different patients. And therefore, using this approach, we seem to be enriching for functional antibodies. Another thing is when you generate, like purely generate AI antibodies, it's not natural to human. Therefore, when you give it to the patient can actually generate the antibody against your drug, and then your drug antibody will no longer be active. Because the antibody we get are from patient already, they are 100% natural, like it's fully human. Therefore, we believe that it will unlikely to be rejected by human. And we also already have a clinic already have a molecule in the clinic compared to other companies who are just providing service to other pharma. So this kind of ends the platform part of the talk, and I will now explain the lead program that we're developing. So this is a novel target that we discovered at a company called IGSF8. And so the way we discovered this is we were asking, oh, if PD-1 is good and it's better than PD-L1 and other targets, are there some similar things like PD-1? There are like pandas in the tumors. Many patients make a little bit of this antibody, and yet it has very little patent coverage. So for each target, we check how many antibodies have people filed patent against this target. And this one is like empty, nothing available. So it was kind of interesting. And IGSF stands for immunoglobulin superfamily. It's a big family of proteins on the cell surface. And all the current immune checkpoint molecules are targeting IGSF, like PD-1, PD-L1, CTLA-4, they all belong to IGSF family. So we ask, are there other molecules like this? Because IGSF family are known to mediate cell-cell interactions. And so we saw this gene, having pretty good antibody signals. Then we ask, how about CRISPR screens, right? We have some in-house CRISPR screens and there's public CRISPR screen. At the time, the Broad Institute has generated CRISPR screens on hundreds of cancer cell lines. And we saw that normally knocking out this gene does not regulate cell growth. The cells are fine, unless the cancer cell is under immune pressure. So we have some in-house data to show that even though it doesn't normally regulate cell growth outside the body, once you implant this cancer cell into the mouse, this is a fully immune competent mouse, then knocking out this gene will make the tumor grow slower. Another experiment is a co-culture experiment. We knock out different genes in cancer cells and co-culture the cancer cell with NK cells, natural killer cells. Again, we saw that knocking out this gene makes the NK cells kill the cancer cell much, much better. So we saw, oh, this might be an immune-related molecule. And then we look at some public data on this. And this is TCGA. Blue is normal, orange is tumor. And so you can see this gene is highly expressed in many, many solid tumors compared to the normal. On the right, in the middle panel here, are cancer types where TCGA has less than 10 normal samples. So you can't really calculate a good differential expression. So we only look at the tumor. It's also pretty highly expressed, especially highest expressed in melanoma. On the right side, there are two cancer types where IGSF-8 is expressed lower in the tumor compared to the normal, and both of them are kidney cancers. So if you work on cancer immunology or immunotherapy, you would know kidney cancers are somehow always different from other solid tumors. In fact, the PD-1 response biomarker is also opposite in kidney cancer compared to the other cancer types, which is really strange. But this kind of gives us also more confidence that this IGSF-8 might be immune-related. We also look at what other signals are correlated with IGSF-8. And so we see that when IGSF-8 is high, the tumors overall have lower CD8 T-cell infiltration. So it's an immune cold tumor. It has lower granzyme and perforin. So the overall tumor immunity is lower. It has lower PD-L1 expression, meaning that this tumor is unlikely to respond to current PD-1, PD-L1 antibodies. It also have very low B2M expression, which is kind of low antigen presentation. This is actually quite interesting. And the last four factors are autophagy factors known to degrade MHC class 1 at the protein level. So, in order for our immune system or T cells to recognize the cancer cell as non-self, the MHC needs to present the mutation onto the surface. But if the protein that express this gets deleted or get degraded, then the mutations are invisible to your T cells. And so we can see basically whenever the tumors are not efficiently presenting the antigens, that's when the tumors have high IGSF-8. In terms of survival, we separated every cancer type, patient half-half by their MHC class 1 high versus low expression. And in 6 cancer types, we saw that when the MHC class 1 is low, high IGSF-8 is associated with worse survival. So when the patient have high IGSF-8, they don't do as well. In other immunotherapy trials, we saw also kind of interesting phenomenon. This is a study by Tim Chan's group where they treated melanoma patients with PD-1. And they have both pre-treatment and on-treatment data. And you can see the benefit patients, which include complete responders, partial responders, and stable disease patients. When they respond, the tumor have lower IGSF-8, whereas the non-responders, the progressive disease patients, their IGSF-8 levels are kind of going everywhere. We also saw another study by Tony Ribas's group, also melanoma patient treated with anti-PD-1. Even from the pre-treatment sample, we could tell if the patient have lower IGSF-8, they are more likely to be having better response to PD-1 and better outcome. So this also gives us some evidence of survival benefits. And so we decided to test this antibody predicted from AI, and we got a very good antibody. This gene is highly conserved between human and mouse, 90% evolutionary conservation. And we are able to use AI to get an antibody that can cross-react to mouse, rat, monkey, and human IGSF8 with similar affinity. So we can use the same antibody for animal experiments as well. So we tested this antibody against, in this case, is a mouse cancer cell co-cultured with mouse NK cells. And if we add different doses of this antibody, we can see adding this antibody makes the NK cells kill the cancer cells better. So in vitro, we also see something. Then we tested the animals in vivo. This is, you implant the cancer cell into the mouse to grow a tumor. This mouse has full immune system, and it's receiving this tumor as if this is an organ transplant. And so when the tumor grow out, we add this antibody, and then some tumor would kind of actually disappear. Other tumor will still be there. Then we can collect the tumor, and we do FACS sorting on the tumor to look at the immune cells. And we saw that with the antibody treatment, the tumor has significantly increased the dendritic cells and the natural killer cells, and also some increase of CD4 T cells and CD8 T cells. And in this tumor, even if we start the treatment after the tumor grow out, after 4 doses, some of the tumors would completely go away. And even if we stop the treatment and wait for another 30 days, the tumors don't really grow back. They are still tumor-free. And this is quite exciting. Our advisor, Gordon Freeman, who discovered PD-1, PD-L1 interaction, said they were not aware of any other monoclonal antibody against this B16 model that can generate a complete response because B16 has a known antigen presentation defects. And so other checkpoint antibodies it has marginal effect, but actually our antibody can totally cure the tumor. And so we decided to test more models and also try compare with anti-PD-1. And so here I'm only showing you data on melanoma and non-small cell lung cancer, but we have also tested CD26 colon cancer, EMT6 and 41 triple negative breast cancer, and we saw very similar result. And in each case, you can see the blue is control antibody, red is anti-PD-1, green is anti-IGSF8, and the purple is if we have the dose of each drug and combine the two like a combination treatment together. And you can see in each case, actually our antibody has monotherapy efficacy in controlling tumor growth. And the antibody works the best when we combine them together. So we did RNA sequencing of the tumor and then just look at the RNA-seq expression signature. We always see the antibody increase NK killing, increase antigen presentation, increase cell adhesion, and increase T cell signaling. And so actually when we saw the animal data, we didn't have any idea what this drug is doing or what this target was doing. The mechanism was kind of figured out over time. And this is the current understanding of this target. In order for PD-1, PD-L1 antibodies to work, the cancer cell needs to present its mutation through MHC onto the cell surface. Then T cell receptor will say, well, there's a mutation. This is non-self. It will kill the cancer cell. PD-1, PD-L1 normal interaction is to make the T cells kind of tired or dysfunctional. And so when we have an antibody to block this, then the T cell can maintain a very active killing state. However, a major mechanism of anti-PD-1, PD-L1 resistance is loss of antigen presentation. And this could happen at the DNA mutation level, RNA lower expression, epigenetic silencing, or protein degradation level. Every possibility, there's actually over 2/3 of the tumors have some kind of antigen presentation defects. And so the mutations are no longer visible to the T cells. Then T cells don't have a chance to recognize the mutation, infiltrate the tumor, or get activated. Even in the tumors that initially respond to PD-1, when they later develop resistance, when people sequence the tumor, again, 50% of them have mutations on this MHC class 1. So it's a major mechanism of immunotherapy resistance. However, our body has a mechanism against this. Natural killer cells are specifically supposed to recognize any cell with antigen presentation defects and kill them, especially if the cell also is they release some stress signals, and if they don't express MHC, it's a very good indication the cell is under viral infection. And so normally NK cell would just kill them. And so then we wonder, well, in the tumors, the cancer cells are under stress, they lose MHC class 1. Why don't NK cells kill them? And we found that, oh, cancer cell is highly expressing this IGSF8. And it was only later we found that it has a natural killer receptor and a dendritic cell receptor. And the antibody that we derived from the patient tumors really block these interactions, both interactions. And therefore, what happens when we dose the patient, or like the mouse tumors, at least in this case, is when you use this antibody to block the IGSF8 interaction with NK cells get activated. They will kill the cancer cells that lost MHC class 1. And if the loss is kind of a soft loss, like epigenetic silencing or gene expression, lower expression, the cancer cell will now have to try a little harder to present their antigen. So we will see the cancer cell kind of increase their antigen presentation a little bit. In addition, the antibody also have a different receptor on the dendritic cells. And this antibody can activate the DC. And so dendritic cells' role is to do antigen presentation, both class 1 and class 2. And also dendritic cell and NK cells, they really help each other active, keep each other active. And once you have increased antigen presentation, both from the cancer cell and dendritic cell, then the T cells get activated. And some of the tumors, when they don't grow big enough, the T cells, when it's activated, is enough to cure the tumor. But then if the tumor is too big, eventually the T cells will get tired. Then you add the PD-1 antibody, they will really help the T cell maintain the killing as well. That's why the two really have synergy. Right? Also, for the tumors that initially respond to anti-PD-1, if we add this antibody to block the IGSF8, then the cancer cell cannot afford to lose MHC to develop resistance. So we also think that the drug will make the PD-1, the cancer respond to PD-1 a little longer time. We have additional data on genetic evidence of IGSF8 as an IO target. I won't have time to discuss this. And so this antibody, because it's derived from human in the CMC development process, this is you go to a bioreactor to make a lot of this antibody, make it really clean, purify it, and put it in a bottle that you can give to the patient. We actually had a very smooth manufacturing process, and we got excellent yield and excellent developability without any optimization at the CDMO company, which is quite unusual. This antibody is like really good. We also did safety studies in rat and monkeys, and the pharmacology, metabolism and safety of the drug in animals also are excellent. And so we started the phase 1 study in patients with solid tumors in March of this year. And the last Friday, we had the safety review meeting of the highest planned dose cohort. So the drug so far, it seems there's no safety issues. So we have a lot of confidence, going to the next stage of the clinical trial, where we'll focus on specific cancer patients. And also, as soon as they finish what's in the guideline, they will be treated with our drug. And we're hoping to see some efficacy at that time. And so far, all the checkpoint inhibitors are developed against T cells. Nothing has been developed in NK cells, dendritic cells, and the macrophage, or at least get approved. There are drugs in the clinical trial. There is some difficulty in developing drugs in the innate immunity because many genes on NK cells, macrophage, or dendritic cell are different between human and mouse. And so when you develop a drug, it's hard to really do this in vivo testing. Also in human, they could also be polymorphic. The KIR, they're very polymorphic. Whereas, we have this one target, it's evolutionarily conserved, and yet it can interact with both the NK receptor and dendritic cell receptor to suppress both cell types. We think it really has a lot of potential. It could be that in the future, all the other immuno-oncology drugs are going to be combined with either anti-PD-1 and our drug, as the combo. So we think it could be the next big checkpoint. And so with our technology platform, we're not just developing this one drug, we're also doing discovery of other targets, all having potential to be first in class. Unfortunately, we don't have enough funding to pursue all of them. So we are now in also discussion with pharmaceutical companies to see whether they are interested in giving us money to do the discovery together. And so I hope this can convince you the company is really at the intersection of high throughput genomics, CRISPR screens, big data, genomics and antibody sequence, and cancer immunology, immunotherapy. We're actually getting a checkpoint. We're combining the power of genomics and AI to unlock the hidden antibody signals already in the patient tumors to develop therapeutics. The first drug is a monoclonal antibody, but recently antibody drug conjugate, radioligand therapy, bispecific antibody, or cell therapy, they all require a tumor-specific target and antibody to bring this drug to the tumor. And so that's an area we think we have some special expertise to identify novel targets and antibodies. What we envision could be a long-term thing, maybe 20 years from now, is some cancer patient come in, we do their tumor sequencing, and based on the analysis, we design an antibody cocktail, multiple antibodies, and we encode them in RNA, and then put them in a liquid nanoparticle, like a Moderna, like you inject that into the patient, and the patient only cells take up the RNA and produce the antibody cocktail to cure their tumor. I think that would be quite exciting. So in summary, I hope this convinces you that tumor, either bulk or single cell RNA-seq profiles are rich sources of therapeutic antibodies. And we are using AI to really decode large cohorts of cancer patients' tumors' B-cell response to unlock the novel targets and antibodies at the same time. We got this antibody to even work in the animals without understanding a thing about this gene. And so this is a really novel innate immune checkpoint in the MHC deficient tumors. And this target has receptors on both dendritic cells and NK cells to suppress DC and NK cell function. And so we developed this antibody to activate the tumor dendritic cell and NK cells, turn the immune cold tumors hot. In many animal models, we see single agent efficacy. And also this drug is synergistic with anti-PD-1. And we also so far has run this safety clinical trial without any safety issues. So I want to thank the people in the company. This is definitely a teamwork. We have a big research site in Shanghai. The headquarters is in US. We also have a number of advisors. And also, since the trial is in the clinic, I want to thank the clinical PI and the patients for giving us the chance. And so I think this is it. Also at the end, I closed my lab last year, but fortunately, because many former postdocs are in faculty positions, they are still maintaining the software in the lab. I'm hoping that these tools will still continue to benefit your research. Thank you very much.
Actually, there. Oh, thanks. That was a really interesting talk. I have a couple related questions. So the first one is, when you get the sort of unmappable antibody mRNAs, is there, there can be errors in sequencing, and I just wonder if, do you essentially rely on the sequence to be perfect, or do you need to sort of do some sort of error correction on your own?
We don't have to do error correction, because even in the same patient, when the B cells are expanded, they go through a somatic hypermutation process to have mutations on the antibody to gradually evolve better antibody. So even from the same patient, you will see different sequences as well. They are kind of different from the germline. But then if we are seeing this antibody repeatedly from patient, we can kind of know what's the right sequence to use. And also, after we get the antibody tested, we do a little bit of optimization as well.
Oh, yeah. Okay. So yeah, that was the other thing I was wondering. So when you have to modify this so that it has this cross-species response, do you, how intensive is it to test different candidates of the cross-species antibody?
So it depends on the target. There are some targets that we're only getting the cross-human, but not mouse. But for IGSFA, we were very lucky. The initial molecule already binds to human and mouse with equal affinity. Then we didn't have to do the affinity maturation just to do the species cross-reactivity.
No,
Yeah,
that's
it was very
amazing.
lucky.
Thanks.
Any questions on this side before I give it to Chengxin?
Thank you so much for the talk. It was very interesting. So I was wondering, you mentioned that single cell sequencing is also employed in conjunction with bulk sequencing. So in single cell, you're able to identify the B cells. So do you, for example, get the reads from single cell sequencing, make pseudo bulks, and then assess what antigen are present in the B cells, or just all of the sequencing together?
So usually if you sequence a tumor, the cancer cells versus immune cells are separate in two separate runs. We can get the antigen data as well from the cancer cells, but then the immune cells, we will be able to run this trust algorithm to assemble the antibody sequences. They are still, I don't think they are really kind of helping to train the model. The data for single cell is way too few compared to what we get from bulk. So most of the training power comes from the bulk tumors. Single cell, it's just nice that the heavy chain and light chain are already paired, so it's easy for us to synthesize that antibody for testing.
Thank you.
Let me read a question that came in the chat. Russell Ryan is asking, for bulk RNA-seq data, how do you match heavy chain sequences with the corresponding light chain sequence from the same B cell?
Yeah, that's a very good question. Right now, if you look at the current AI field for matching heavy chain, light chain, they have some power in naive B cells in the blood, but they have no power once the antibody started a clonal expansion. For us, we are getting the bulk tumor. So initially the heavy chain and light chain are not matched. We do, for a lot of heavy chain, we do kind of think maybe multiple light chains are possible. But if this antibody is repeatedly expanded in many patients, you would see the same heavy chain and light chain in many patients. We do see a significant enrichment of that, which can help us narrow down the choice to test. But it's certainly not perfect yet. That's why single cell data is very helpful for us.
Yeah, so in your AI model, you don't use any specific antigen antibody ground truth labels, but there's actually a small number of antigen antibody pair that we do know the exact label. So do you think spiking in those antibody antibody data with explicit antigen labels could potentially improve the accuracy of your AI model?
Yeah, that's a good question. It depends on how that antibody is generated, because some of these are generated in mouse or camels, and then they humanized. I don't think they will follow the same rule as a human antibody. But if it's from panning, like phage panning, but it has to be a synthetic library from, well, no, not from natural human B cells for a panning library, maybe it will help. But again, it's a very small subset compared to the 100 million antibody that we're getting from tumors. We can use it to kind of help us validate, but we haven't, I don't think it's going to sway the training as much.
Shirley, I have two more questions coming in online. Yuan Fang has two. Let me read her second question first. You mentioned high affinity, yet no safety issues in phase 1 trials. Yuan Fang is asking for popular PD-1-like target, such as Keytruda and Atezo. Adverse effects, especially ADA, always affect a large fraction of patients. Is it not seen in your case?
So right now, we have not treated our patient enough time. In order for ADA, you need a longer time treatment. So for our safety, it's a 21 cycle. And then if it's safe, at this stage, what we care about is patients don't develop cytokine storm and die or have very severe side effect. And we don't see that, at least in the first 21 days. In order to see whether there's ADA, it's going to be much longer time. But so far, if you look at the AI companies or pharma trying to develop antibodies without immunogenicity or the likelihood to generate ADA, their main goal is to make this antibody look as human as possible, which naturally our antibody is human already. Yeah.
Any other questions? Okay. While I'm walking over, I mean, say Yuanfang has a technical question on one of your survival plots, and if there are 10 mice or fewer, how did you get the p-value of maybe
So you mean this one?
18 on the slide?
So each arm for this, we have 8 mice, but we have done many different models, many different conditions, and also CD4 depletion, CD8 depletion, NK cell depletion, different combos. So we are fairly confident about the animal data. I think overall the different animals we're talking about, I'm only showing you a subset. We have like probably close to 300 animals for the in vivo data.
Have you ever looked at the maybe PD-1 or in your case like IGSF8 antibody resistant clone or something like that? And then do you see like the kind of evolution with the antigens that existing in the tumor cell acquire additional mutations in the antigen so that the antibody you design actually also like the escaped from the tumor cells?
So far, we haven't seen much specific like point mutation on IGSF8, but this gene is frequently amplified in the tumors. We think that's also a potential evasion mechanism. So this gene normally is expressed in brain and neuronal tissues because our brain, the neuronal cells don't regenerate. And so no matter what mutation happens, you don't want your immune system to attack this. And so neurons don't express MHC class 1, and that essentially suppresses the adaptive immunity. And they also highly express IGSF8, and that will suppress the innate immunity. And we hypothesize the cancer cell just kind of learn it from the brain as well when they lose MHC class 1.
Thank you so much.
Thank you. Very nice talk. I have one question. So you can deduce the antibody sequence from the public data. So how do you find all the antigen from public data?
The antigen is in the tumor profile as well.
Oh, this is a protein sequence or?
There are, RNA sequence and also other like mutations and other immune signals that we are considering in that as well. We also do our additional annotation on the tumor as well.
Oh, okay, thank you.
But it's all there.
All right, maybe I'll take the honor of asking your last question. Shirley, could you comment more about the tumor type specificity, like which types will stands to benefit in the near future?
So, PD-1 has been approved in 18 different indications, but in each indication, only a minority of patients benefit. And so now we are doing this trial. We are kind of planning for the next stage when we have to settle on a specific cancer type. What we are looking for is some cancer types. Right now, we cannot use IGSF8 to select a patient because FDA needs a retrospective study to show that patient with higher IGSF8 will benefit more from the drug before we can use it as a biomarker for patient selection. What we can do is to ask which cancer types enrich for higher IGSF8 in general, like melanoma, enrich for MHC class 1 loss, which is like lung cancer and colon cancer, and also which cancer types have unmet clinical need. We ask the clinics, do you see patients, what line of treatment can we get? So normally for each additional line of treatment, the patient gets sicker and they have waited even longer time. There's usually a 10% drop in their response to immunotherapy. So we want to go to as early line of treatment as possible. So we are asking, is this disease very crowded? Too many other investigational drugs being tested, then we don't go to those. So it's a kind of complicated decision. Also fairly frequent in the US, we can get enough patients. That's a kind of holistic consideration.
Good. Complicated decision. At
Yeah.
end note, please join me in thanking Dr. Shirley Liu